So we've talked through the steps of the sliding filament model of contraction. We've talked through this process that shortens sarcomeres, shortens myofibrils, shortens muscle fibers, and shortens or contracts muscles. This is a process that also goes by the name excitation, contraction, coupling. It's the coupling of the nervous system with skeletal muscle, causing that muscle to contract. Now, I've put together all of the steps that we've talked about so far, from the time the brain sends out the impulse to the time that all the calcium is pulled out of the muscle fiber and relaxation occurs. I've put all those steps together into a handout for you. You can use that handout while you're studying. But now I want to move forward and talk for just a couple minutes about the two other kinds of muscle cells that we find in the human body: cardiac muscle and smooth muscle. Cardiac muscle, like the name suggests, is found only in the heart. The cells that make up cardiac muscle can be called cardiac muscle fibers, but they're also known as cardiomyocytes, heart muscle cells. When you look at cardiomyocytes under the microscope, the first thing that strikes you is that they are much shorter than skeletal muscle fibers. They also have a single nucleus, and it tends to be centrally located in the cell. They are striated. They do contain sarcomeres. But unlike skeletal muscle fibers, cardiomyocytes are branched. They are bizarre looking cells, and I'll show you a picture of this in just a moment. Each one of the individual cells is connected to other cells, and all the branches are connected to each other with structures that are called intercalated discs. Cardiac muscle cells do contain myoglobin, so they can store oxygen like skeletal muscles can, but they don't contain any sarcoplasmic reticulum. They are much smaller than skeletal muscle fibers, and so they don't need a ready stored supply of calcium. Instead, they have calcium pumps located in the sarcolemma, and when the time comes, they simply pump calcium in, and calcium then regulates the contraction of the muscle cells. Cardiac muscle cells, cardiomyocytes, have a single job to do, and that job is to provide a very coordinated wave of contraction throughout the pump that is the heart. The heart does not contract in unison, it contracts in a wave so that it pumps blood throughout the whole body and delivers oxygen and removes carbon dioxide from the tissues. On this slide, we're looking at a drawing of cardiomyocytes, and we're looking at an actual image taken under a light microscope. If you start over here on the right side, you can see that right away these cells look different. Remember how big the skeletal muscle fibers were? These are much smaller. They're just smaller overall. The other thing you notice is that they have branches. These cells actually send out branches of themselves. Very, very strange looking cellular anatomy. Over into the diagram, you can see individual cells that are all joined together. So there's a part of a cell here connecting to this cell. Here's the nucleus of the cell. This cell is branched here and here. It's meeting another cell here. Here's another nucleus. Here's another cell and another cell and another cell. All of these cells are connected to each other. They're joined together at these structures called intercalated discs. Now, I should note here that it is possible for cardiac muscle cells, cardiomyocytes, to have more than one nucleus. It's just not common. They develop differently than the skeletal muscles do in the fetus. The intercalated disc that holds cardiomyocytes together has two jobs to do. Number one, it physically anchors those two cells together. That's an important job. And number two, it creates an electrical synapse between the cells. Remember, when we talked about electrical synapses, we said that they were composed of gap junctions that allow for the electrical signal to travel from cell to cell with very little delay. So intercalated discs, which are embedded into the sarcolemma, provide those two key structures: gap junctions that create the electrical synapse between cardiomyocytes, create a tunnel, essentially, for the electrical signal to travel, and desmosomes. Desmosomes are simply connection points, anchoring points between two adjacent cells. So they're held tightly together and they are connected with electrical synapses. Now, the other note I have here is that the gap junctions, the intercalated discs that occur between cardiac muscle cells, form another unit of contraction. I said a moment ago that cardiomyocytes don't all contract in unison in the heart. They have to contract in a wave pattern across the heart. It turns out that it's groups of cardiomyocytes that are contracting together in unison. And when cells function together in unison like this, we give that group of cells a name. We call that a syncytium. A syncytium of cells that are going to contract in unison. So one syncytium followed by another, followed by another, followed by another, causes the wave of contraction across the heart. Smooth muscle cells are very different. Smooth muscle cells are short and much smaller than skeletal muscle cells, but they don't look anything like skeletal muscle cells. They are spindle-shaped. And again, I'll show you a picture in just a moment. Like the cardiomyocytes, they tend to have a single centrally located nucleus. But unlike cardiac cells, unlike skeletal muscle fibers, smooth muscle fibers are non-striated. They do not have sarcomeres in them. So skeletal and cardiac are striated, smooth are not. Smooth muscle cells have no myoglobin in them, so they cannot store oxygen the way the other two types can. They do have sarcoplasmic reticulum, but it's very poorly developed, and it's really not used as a storage place for calcium. Instead, similar to the cardiac cells, smooth muscle cells just pull calcium into the cell from outside using calcium pumps. Smooth muscle provides for involuntary muscle contraction. Smooth muscle tends to be located in the viscera. It's located in the walls of the hollow organs throughout the body. It's located in passageways throughout the body, things like the bronchi of the lungs, things like the ureters in the urinary tract, things like the reproductive tract. It's located in the walls of the stomach and the intestines. It's located in places in the body that are under autonomic nervous system control, involuntary nervous system control. And because of the fact that it provides contraction for viscera, it's often providing long periods of contraction with very little rest. The reason that smooth muscle can achieve such long periods of contraction is because it also achieves a much lower level of force than skeletal muscle does. So smooth muscle has no striations, no sarcomeres. But interestingly enough, it still uses actin and myosin for contraction. It still uses thin and thick filaments. They are just not organized into sarcomeres. The other thing to know is that smooth muscle uses intermediate filaments as well. And I'll show you a picture of this in just a minute. The myosin, the heads on the myosin molecules are going to create cross bridges with the thin filaments. And the motion of the myosin heads is going to slide the thin filaments, but not in sarcomere arrangement. The contractile activity of the thick and thin filaments is not regulated by troponin and tropomyosin, but instead by a protein called calmodulin. The intermediate filaments are going to create a meshwork around the interior contents of a smooth muscle fiber, just underneath the sarcolemma. And those intermediate filaments that create that mesh, they're going to be anchored to the sarcolemma with structures called dense bodies. Let's take a look. On this slide, you're looking at a drawing of two smooth muscle cells, or at least a portion of two smooth muscle cells side by side. There's one here, and there's one here. Notice that these cells have sort of a spindle shape. They start off quite narrow, then they enlarge. If we could see the rest of the cell, it would do the reverse. It would come back down into a narrow spindle shape. Now, the sarcolemma has been taken off of these two cells. We're looking at what's right underneath the sarcolemma, and that's this strange net of fibers all around the interior of the cell. The black, those are intermediate fibers. Where you see green, those are thick filaments, myosin. And where you see orange, those are thin filaments. The pink regions, those are the dense bodies that are attaching this mesh to the sarcolemma, to the inside of the sarcolemma. Now, calmodulin is not shown in this image, but calmodulin is going to interact with calcium. And what's going to happen is, in the regions where the thick and thin filaments are, the myosin is going to move and slide the thin filaments on top of itself, contracting or shortening this region. So the thin filaments in orange are going to slide over the thick filaments in green. That's going to shorten the distance between two dense bodies. Those shortening actions are going to pull on the intermediate filaments, causing that entire meshwork to tighten around the cell. So you start with the cell fully relaxed up here, and in the presence of calcium and calmodulin activity, you're going to get this. The whole meshwork is just going to tighten around the whole circumference, the whole length of that cell, all at once, causing contraction. Here's another image. I like this image because it really drives home that you've got this elongated spindle shape when smooth muscle cells are relaxed, centrally located nucleus, and then this meshwork of fibers on top. Some of these are intermediate filaments, Some of these are thick and thin filaments. This term calveoli, don't worry about this term. I'm not going to test you on it, but just so you know, when the calcium is pulled in through the calcium pumps in the sarcolemma, it tends to pool in these regions called calveoli. Don't worry about that term. When calcium floods into the cell and binds to the calmodulin, it's going to trigger the myosin heads to start forming cross bridges with the thin filaments. The myosin thick filaments are going to pull the thin filaments on top of themselves. You're going to get a shortening of that area, that region of the net. It's going to pull on the dense bodies, and the dense bodies are going to pull on the intermediate filaments, and this is going to happen. The whole network just gets tighter, and that basically pulls that whole anatomy of the smooth muscle cell into a smaller space. It contracts it. This is all happening underneath the sarcolemma of the smooth muscle fiber.